Well, I know that people are still coming, but I think we should get started. So it's a great pleasure to welcome you, Bill, here, and really looking forward to your seminar. So Jane just gave me a 40-page CV of Bill, so I'm facing quite a challenge here. So I'm going to shorten it. There is a very nice summary of Bill's achievements on the ISMB website, because it's also a good time to congratulate him on the ISMB Innovator Award that he just received. It was just announced a few weeks ago. I guess you haven't received it yet, but it's coming. So using that very short summary that they posted, so Bill Noble is a professor of genome sciences at the University of Washington, and also professor of computer science as well. And he received his PhD degree in, well, less than 20 years ago, 1998, in the University of California, San Diego, and then did a short postdoctoral fellowship in David Hausler Lab at the UC Santa Cruz, and moved to New York to Columbia University as assistant professor, and then moved to the University of Washington 3 years later, and remained there since that time. So Bill is really internationally recognized for his work on developing machine learning methods for all sorts of high throughput data, from proteomics to genomics, including structure modeling. And today we'll hear his recent work.
Thanks, Alexei, and thank you all for coming. I've enjoyed visiting with people here today. There seems like there's a lot of exciting stuff going on. It's also nice to be back in the Midwest. I grew up outside Chicago, so I think of Michigan as the place I always went for vacation. So it's nice to be here. I'm going to talk today about a couple different projects. As Alexei mentioned, my background is in machine learning, and I've made a career out of sort of trying to apply machine learning methods and develop new machine learning methods for making sense out of biological data. So I've got a collection of 3 different projects. I'm actually going to focus on the first and the third and just briefly mention the second one. And they all sort of share one aspect, which is that there's a latent space that's involved, which realistically, it's not like I went in thinking, oh, I've got to find different ways to build latent spaces. It was more like afterwards I looked back and said, oh, look, these projects we're doing are kind of similar in this way. So I'm going to dive in by telling you a little bit about a consortium that I've been involved in since its inception in 2003, and probably most of you are familiar with. Encode stands for the Encyclopedia of DNA Elements, and it was meant to be a follow-on to the Human Genome Project, where the Human Genome Project sort of gave us the book, and Encode is supposed to help us read it, right? In the sense that Encode is trying to find what are the things in the genome that actually do stuff, the functional elements. Encode is in many ways just a data generation consortium, but the idea is not just to generate the data, but also to interpret it. And the kind of data that's being generated essentially measures different kinds of biochemical activity along the genome. So you have, for example, measures of gene transcription, most notably RNA-seq measurements. You have measurements of protein binding, like transcription factor ChIP-seq. You have measurements of DNA local packing density, DNase, or ATAC-seq measurements. And all of those together are carried out in many different cell lines and tissues. So this is just a picture from another consortium, the Roadmap Consortium, showing you sort of the variety of different tissue types that you can imagine doing this kind of experiment in. But of course, the space is even larger than this, because you can have lots of different perturbations. You can do CRISPR manipulations of cell lines and so on. So you have a large space of possible kinds of cells, and then a large space of possible measurements to perform in each of those cells. If you're thinking of this like a computer scientist or an electrical engineer or a statistician, it's helpful to say, well, okay, there's all these assays, but realistically, they are all involved short read sequencing, and you essentially summarize everything as a measurement along the genome that has to do roughly with how many sequences covered each base of the genome, so you can represent it all as just one vector. It's a long vector because the genome is long, But this is just a sort of showing a short piece of 50 kilobases. Well, that bar is 50 kilobases of signal for 3 different assays. So these are measurements of histone modifications, 3 different kinds of histone modifications in 3 different cell types, where the value at each position shows the level of modification as measured at that position. So the point of the first project is that this is a big space of possible data. So there's some types of assays that were performed sort of systematically across many, many different cell types. And then there are some popular cell lines that were characterized in great depth with many, many different kinds of assays. But there's a lot of purple in here also, which are all of the assay cell type combinations that have not yet been performed. So this slide is actually old. This is from the original data set that we performed our initial analyses on. So when I say it's big, it's true, it's big, but it's bigger now. So there's actually more than 10,000 experiments in Encode currently. But this is a picture of sort of a chunk of the data where you can see in yellow the experiments that have been performed, and purple the ones that have not yet been performed. And about 5% of this matrix is complete, or at least of the full matrix is complete. So the problem is, if you are a particular biologist studying a particular kind of cell type, and maybe you're interested in a particular transcription factor, you go to encodeproject.org and you say, all right, I'm going to go get all this data that the taxpayers paid millions of dollars for, and it's going to tell me, does my transcription factor bind there, or is there histone modification occurring at the position that I'm interested in the genome? And lo and behold, they didn't bother to do it. Now you can email the Encode investigators and lobby to have them do the experiment, or you can do it yourself. But maybe that particular type of cell is hard to get, or you don't have the facilities to do it. It would be nice if you could try to use a computer to predict what would happen if you did do that experiment. So that was the project that Tim Durham tackled. So Tim is a graduate student in my lab, and he worked together with Max, another graduate student, Jeff Haubert, who's a research scientist in the lab, and Jeff Films, who's in electrical engineering. And the project was motivated in part by Jeff Haubert's participation before he started in my lab in the Netflix Prize Challenge. So Jeff pointed out that recommender systems have been extremely successful in many different settings, and in particular, they were useful in the Netflix challenge. So this was a challenge in which Netflix handed out a database of movies and users and asked people to predict, here's a user, I'll give you some of their preferences, now predict what other kinds of movies they like on a scale of 1 to 5. And the idea is that, let's say that Tim is a particular user, and this is a particular movie. You'd like to, in a matrix decomposition approach, you decompose this matrix into 2 matrices, each with a latent dimension of k, k values, where the row here represents sort of features of the movie, and the column here represents features of the user. So you can say, for instance, this value might represent how much is this movie about cycling, and this value might be, does Tim like movies about cycling? And as you can tell from his sporty gear, he does like cycling. And so then you can predict that he's probably going to like this movie. So the idea, relatively straightforward idea in this first project, was to try to do this, but use, instead of a matrix, use a tensor. So the tensor version of matrix decomposition was described back in 1970. It's called Parafac. But the basic idea is exactly the same as what I just described to you, except that instead of movies and users, we have cell types, assay types, and genomic positions. And so what you get are these 3 different sub-matrices, or the different matrices that are produced during the decomposition. And when you take a sort of generalized scalar product of the corresponding rows or columns from each of those matrices and combine them, then that gives you a prediction for a particular cell type, a particular assay type at a particular position in the genome. The optimization, this is the original Parafac paper. The optimization looks like this mathematically. It looks a little daunting, but it's actually relatively straightforward. All you're doing is summing over all the different possible combinations that I just described and saying how close, what's the difference between the value I get when I predict at this position, and the actual value. So here's the actual training set value, and here's the value you get when you do this scalar product with some additional factors built into it. And you take the mean squared error between those. And you put in some terms to do some regularization to prevent overfitting. This turned out to be challenging for many reasons. In particular, it's a fairly large data set, and so we wanted to do it in the cloud. So there was a large part of the paper about this is about how you get this to work in a cloud computing setting. And we did it both on Amazon Cloud and Azure, mostly for logistical reasons having to do with both funding and getting around bugs. But it ended up working quite well. Now, I'm not going to show you all the results there because I want to focus on the next project, which goes a step beyond this. And I'll convince you that that one worked even better, and that's the sort of the key take-home. Question? So no, it's funny. Tony and I were hired at Columbia like basically the same year. So I know Tony, and we co-advised the student, but I don't know anything about what you're talking about. Yeah, I'd like to hear. Okay. Oh, really? Okay, great. And I meant to say, actually, please do interrupt if there's other questions. I'd rather stop and get clarifications and so on than power through and lose everyone. Yeah, yeah. I see. Interesting. Okay, so what happened next was that Jacob Schreiber joined my lab, and he developed a piece of software. All of his software is named after fruits, and avocado apparently is a fruit, even though I kind of think of it as a vegetable because it sort of shows up in your salad. And so he developed a software called Avocado that is sort of the predicted version 2.0, and this was done in collaboration with him and Jeff. So the basic concept here was, first of all, Jacob likes deep learning, right? So he's coming into it with that sort of prior, right? He's like, okay, I want to find some way to apply deep learning, but we already have this successful infrastructure. The key drawback, or one key drawback to the way that we did this with the tensor decomposition was you're sort of treating everything independently, but more to the point, you're also just doing this relatively straightforward generalized scalar product to combine the latent factors. And maybe that should be done in some nonlinear way. But it turns out there's an existing set of tools called deep tensor factorization that do allow you to go to a nonlinear setting, where you essentially take your assay position and cell type things, multiply them all together, and then take the resulting vector and put it through a deep neural network. And that's called deep tensor factorization. Jacob made a slight modification of that idea with the idea that we don't necessarily want to assume that we have to multiply the values together first. We want to actually allow the multiplication to be generalized also. And so rather than multiplying first and then putting the multiplied values through, he just concatenated them. And this has the nice side benefit that we no longer have to have the same latent dimensionality for the different kinds of factors. So you could have, for example, more factors for cell type than you have for genomic position. And that turns out to be quite helpful. So in particular, for the genomic position, this is where the bulk of our parameters are of this model, because even though we're doing this at we're doing this at 25 base pair resolution, there's still a lot of positions, even if we're only looking at the pilot regions of the genome, which is the 1% of the genome that Encode focused on initially. So the idea is, could we solve or address the independence problem that I alluded to earlier while also reducing our parameter space? And the idea is, well, we know that the different kinds of phenomena happen at different scales. If you're looking for a particular transcription factor, maybe that does bind at the 25 base pair resolution or something close to that. Whereas if you're looking at some nucleosome-sized thing, that's a different, or even histone modifications happen at the level of nucleosomes. Those are more like 250 base pairs. And then you might have gene-sized or larger elements. And so what we did was divide our factors into some that are defined at 25 base pair resolution, some at 250 and some at 5 kilobase pair resolution. And so this reduces the total number of parameters in the model by about a factor of 5. You could ask, how do you come up with 25, 250, and 5 kb? And those were essentially just chosen based on our prior knowledge. You also ask, how did you come up with 15, 35, and 50? That was done essentially through a hyperparameter search. So we have to choose the hyperparameters of the model, how many layers, how many neurons per layer, and then how many factors for each of these things. We have 7 different hyperparameters. And we just do a random search over a grid of these hyperparameters and essentially measure the mean squared error. So this is just a plot of a histogram of all those mean squared errors for different parameter combinations trained on that 1% of the genome. The 2 bars here show the performance of chrome impute and predicted. So mean squared error, right? So smaller is better. And so we chose the model that works the best on the validation down here. So this gives us a performance increase, a 4.4% decrease in mean squared error compared to predicted, and a 17.5% decrease compared to the first method developed in this field by Manolis Kellis and Jason Ernst called chrome impute. This is just an aside to let you know, if you look at the results from that hyperparameter search a slightly different way, you can see that the neural network parameters, the topology parameters, are by far the most important. Each of these plots is just marginalizing over one of the 7 hyperparameters that we were searching for. So these are the 5 different values for a given hyperparameter, and these are the histogram of mean squared error values for all runs where that value was, say, for example, 256. And the main observation is that these are relatively flat, whereas these show a marked trend. As you get more larger models, you're getting improved performance. Okay, so practically speaking, what this means is that you can look at real data like this, and then you can try to impute it, and you can see that qualitatively, all 3 of these different methods seem to perform well in the sense that they capture the main qualitative features of the data. We can tell you that quantitatively that we do have an improvement in the lowest one, which is the Avocado imputation. There's a bunch of metrics that I'm not going to go through, so I'm just going to skip this slide because otherwise you'll stare at it and try to figure out what they all mean. And instead I'll focus on the subsequent part where we tried to zoom in a little bit more to ask what we thought were kind of the interesting questions, which is, if you think about when are you going to use an imputation method like this, what you really want to be able to do is find peaks most of the time. If you're looking for, say, for histone modifications, for example, or even gene expression values, you'd like to be able to predict where there's large peaks in the data. And most of the time, what's interesting to you are peaks that are sort of unusual, right? If you look at 27 different cell types and you see a peak at this position every time, and then someone predicts in the 28th cell type that there's going to be a peak there, you're not going to be super excited. You'll be like, no duh, right? But if it's like half the time peak, half the time not, and you're pretty good at predicting in the next one whether it is, that's a hard problem, right? So what we decided to do was segregate all of the peaks according to how frequently they occur. So here's a picture just showing, a peak is present in this number of cell types. So you have like all the ones that are constitutive, and those get evaluated separately from all the ones that are super rare, and the ones in the middle are sort of the hardest ones to identify. And so we looked at this 3 ways. We looked at the mean squared error separately in each of these different kinds of peaks, and then also recall and precision, where we had an argument in the paper about how we set the threshold for that. And so here's the results. What you can see is that here what we've done, this is supposed to be over there. Number of cell types, these regions are a peak in. So you've got some of these things are constitutive. Some of them are extremely rare peaks that only happen in a few cell types. And these ones in the middle are the harder ones where it's sometimes there and sometimes not. And if you look at the mean squared error, you can see there's an improvement systematically between Avocado relative to chrome impute and predicted. Then we also can do recall and precision. And here you can actually include the real experimental data as well because of that thresholding step that's involved. And what you can see is that in terms of recall, it looks like Chrome impute actually does really well compared to the other two. But in terms of precision, it does much worse. So what this is telling us is that Chrome impute is sort of over-predicting peaks, right? Alternatively, you can think of it as these other two methods are under-predicting peaks. So you're getting a trade-off here, precision and recall, between the Chrome impute method and the two methods that we've developed. And so, of course, that's a trade-off that, as an end user, you'd want to know about and sort of help you to interpret the peaks that get predicted. Yeah, question? It gets easier and easier to say this is just there all the time. The ones that are easy are the ones that are always there or always not there. Okay. Okay, so then the other half of this Avocado project was to say, okay, we've done all this learning. We now have these latent representations. Maybe they can be used in a transfer learning setting to help us do other things well. So maybe those latent factors provide a useful summary. So for example, if you just take the latent factors on the cell type axis and you reduce them, so they were, honestly, I don't remember. Let's say they were 50 dimensional. I can't remember what number we came up with. Let's say they're 50 dimensional. We reduce them to 2 dimensions using TS&E and project here in this picture. And what you can see is that if you color them according to sort of the canonical region of the body, that, for instance, all of the blood cell types cluster together and so on. So that was encouraging. It said the latent factors seem to capture something real about the biology. But for a more quantitative approach, we decided to try to use the genomic factors. These are 110 values for each 25 base pair region of the genome to try to solve a number of sort of canonical prediction tasks in genomics. And so we used a gradient boosting classifier just because that's a fairly robust and widely used classifier. The claim here is not that this is going to produce the state-of-the-art method, but mostly to show that these factors are a useful way to represent the genome. And we do cross-validation across chromosomes in a leave-one-out fashion. And we compare 5 different kinds of inputs. You can either just take the roadmap data directly and put it into the gradient boosting classifier. So this is the data set that we used initially. Or you can take the imputed data. So this would be the roadmap data plus all the imputations, so extending it to be a much longer vector where you have all the imputed values as well. And those can be imputed by any one of these 3 methods. Or you can just take the 110 Avocado latent factors. And our hypothesis is that those are an information-rich way to represent all the information in those other vectors in a way that's sort of amenable to machine learning. So the first experiment we did was predicting gene expression. So what we're doing is we're taking RNA-seq data from the roadmap project in 10 different cell types. And we're inputting 5 histone marks in promoter regions and predicting positions with RNA-seq expression greater than 0.5. So this is a fairly standard way to frame this prediction task as a classification task. And actually, I wrote 10, but that's from an older version of this slide. There's obviously many more than 10 cell types here. And we've just sorted them according to the performance of the best, actually, of the baseline. So the majority baseline is just if you took the average among all the training set cell types and used that as your prediction for this cell type. And for each cell type, you can see all of the different colors here. And what hopefully you can tell is that there's a lot of green up near the top there. Or sorry, a lot of red is what you want. The red is up near the top there. But it's hard to see in the picture, so I put some statistics. Avocado improves over just using the epigenomic measurements, the raw data, in all cell types, and by an average of 0.144. Here, this is the average precision, so averaging over the recall axis. And it even performed better than the full roadmap compendium. This means taking all of the different data, not just from your cell type, but from every other cell type, and putting it into the classifier in 36 out of 47 cell types. So this seemed like a promising evidence that this was a nice way to represent the genomic axis. We also looked at a number of other tasks. I didn't include them all. We have promoter-enhancer interactions, predicting replication timing, and now Jacob's currently looking at predicting exons. But we did, this one was frequently interacting regions. This is a feature of Hi-C data that essentially corresponds to, it often corresponds to the edges of particular kinds of topological domains in Hi-C data. Essentially, the way it works is that you look for sort of a cross-shaped region like this, where you see particular density within these bars, and that means that the thing at the center of the cross is a frequently interacting region. There's actually a statistical model that tries to account for biases to identify these frequently interacting regions. But we just downloaded the FIRE annotations from this paper, Schmidt et al. From Cell Reports, and then asked, can we plug it into our gradient boosting classifier and make predictions? So that's an open question. The question was if there's more than 2 TADs coming together. It's not clear what the fires actually correspond to. I think that is one model, because there's so much interaction happening that there may be lots of interaction. That's right. That's right. For our purposes, all we know is we have a clear definition of them, and so we'd like to be able to say where else might they be along the genome. And so this is showing similar kinds of plots as before. It's the mean average precision, which is a really weird, but I guess a fairly standard term, mean and average. It's average because it's averaged across all the different recall values, and then it's mean because it's mean over different chromosomes or different, I guess it's over different genomic positions. Anyway, I tried to change it to average precision in the other slides and forgot. But the point is that we're again comparing the same kinds of things here, and the Avocado latent factors, in nearly every case, well, in every case they outperform using any of the imputed values, and in nearly every case they also outperform using the full, all the real data from the roadmap compendium. So this is a much more concise way to represent the genome, and it captures a lot that's in there. What you can also see is that if you take the full roadmap compendium, it doesn't do as well as that. This is data from all the other cell types. So you're trying to predict whether there's a fire here by looking at what's going on in other cell types, which is a little bit counterintuitive until you remember that that contains a bunch of other kinds of measurements that weren't necessarily available in the cell type that you're looking at. In particular, CTCF and some of the other factors that are really informative are often not available in the blue bar, but are available in the green bar. So what we're currently working on now, besides making the model available and getting the paper published, is to think of other ways in which this kind of latent representation of the genome can be useful. And so if you have ideas for where this might fit into your research, we'd be happy to hear it. The other thing that we're doing is organizing a challenge, which was announced last week. So this is being organized by myself, Anshul Kandaji, Manolis Kellis, and Ziping Wang as part of the Encode. So this is we've put together sort of a data cube, a tensor of data that's got a lot of missingness. We've got some prize money for winners, and the challenge has 2 different phases, January 28th to May 14th and June 3rd to August 14th. With the idea that once we get all of the experiments done, we would write a paper about the results. A key aspect of this is that we have truly prospective validation, in the sense that data generation labs within Encode have agreed to perform additional experiments which have not yet been done, right? So that when we get the predictions, we'll be able to test them on test sets that really no one has ever seen before. So that's the end of project 1. How am I doing? Pretty good. All right. So I want to briefly tell you about another project with someone who you probably know, because he's sitting right there. But I didn't want to give you tons of detail because he can tell you all about it. But that does fit into the same kind of, some of the same ideas come out of this. So Now we're talking about single cell Hi-C data. This is a project that basically Gia did with some help from Dejun and Gurkhan, who were helpful in making sense of some of the data formats and so on. So having talked to a lot of you, I know a lot of you already know all this. But just to be sure we're all on the same page, single cell Hi-C is a sort of a single cell variant of the Hi-C assay that measures the three-dimensional conformation of DNA in the nucleus. So the idea is you're going to isolate a single cell. You do this ligation, fill-in, restriction enzyme digestion, and cross-linking, and so on. And what you get out is a very sparse matrix in which the rows and the columns correspond to positions along the genome, and values in the matrix correspond to counts of how frequently you observed contacts between those positions. So in this one, each of these boxes corresponds to one chromosome. You have lots of intra-chromosomal contacts in dark, and then the inter-chromosomal contacts are sort of in a lighter shade here with fewer contacts. One of the big questions is, if you look at the RNA-seq literature, a lot of what people are interested in with single-cell analysis is stuff like this. This is the Monocle software produced by Cole Trapnell in our department, and it's trying to reconstruct differentiating the trajectory of development in a population of differentiating cells automatically from the data. So this is a projection down to 2D and then followed by this kind of tracing step. There's other kinds of projection that you can do, just doing things like TS&E. It's just showing tissue type clustering in some single cell data as well. And so the question is, could we do a similar kind of thing with single cell Hi-C data? So one of the things that Gurkhan helped with, I mentioned Gurkhan was one of the people involved here. He had been involved in a previous study run through Encode, in which our lab sort of coordinated 4 other labs who were developing ways of measuring the reproducibility of Hi-C data. So they were measurements for how similar is this Hi-C data set to this other Hi-C data set. And so what we did was said, well, maybe those will work for single cell as well. So what Gia did was took one of those existing methods called Hi-C rep, and then combined it with a standard dimensionality reduction technique, multidimensional scaling, and took some data where this was published data in which we actually had labels for where the cells were in the cell cycle. You can see G1 and cyan, early S, mid S, and late S, and so on. And this was in the sort of test set that we initially pulled out, where we evenly sampled from each of the cell cycle phases. And here's from the full data set. And this might not seem so impressive to you, except if you hadn't tried a bunch of other stuff the way we did, and nothing else gave us much structure, and then suddenly Hi-C rep, that's pretty convincingly cell cycle. Even if I didn't color it, you could pretty much see that there's a circle going on there, right? So Gia can tell you a lot more about this. This was presented last year at the ISMB, but the point is, this is a very nice way to take single cell data and project it down into a lower dimensional space, a latent space in which the relationships in this lower dimensional space now clearly reflect, at least for this data set, the underlying biology of cell cycle variation. Okay, so now I'm going to change gears again. And as promised in the title, I'm changing from genomics to proteomics. I know there are some people here who do proteomics. That's nice. I spend about half my time thinking about protein mass spectrometry. And so I want to tell you about some of the work we've been doing on GLEAMS. So first, just in case some of you don't do proteomics or haven't heard so much about it, just to remind you, the key idea here is that you have some complex sample. Whatever the sample is, it's got proteins in it. And as you know, proteins are complicated looking, right? They've got all these fancy structures associated with them. And biochemically, proteins are kind of hard to deal with, especially in high throughput. So the way that shotgun proteomics works is that you take these proteins and you first cut them up into pieces using something like trypsin. And the trypsin gives you somewhat deterministic cleavages of the proteins. And these peptides are the things that are subject to mass spectrometry. You put them through a device. This is a picture of a device in our department. That's actually sort of an old one by now. And then out comes a bunch of observations. They're spectra. which really are just basically vectors again. And I'll tell you a little bit more about the spectra in a bit. Actually, I'm not going to tell you that much about them, except to say each of these represents canonically one peptide species. And the height of the peaks corresponds to different fragmentation events along the peptide backbone. So then you put it through some fancy computer, and ideally, out comes an identification for each of the observed spectra. You can say, this is the peptide that I think was responsible for generating it. So the project I want to tell you about is GLEAMS. GLEAMS is a recursive acronym. GLEAMS is a learned embedding for annotating mass spectra. That stands for GLEAMS. And it was developed by Damon May, a PhD student in my lab, and also in collaboration with Jeff Films in electrical engineering. So one motivation for this project is that most protein journals require that your data, once you've finished it, that you stick it into a public repository. So some of the common repositories are things like Massive, Chorus, Pride, and so on. But unfortunately for the field, this repository submission is usually the end of the process. So you go along, you generate your data, you analyze it, you write your paper, you deposit your data, publish, end of story. And what would be nice is if we could move this up a little bit more. Genomics is much more advanced in this respect than proteomics, where sometimes you actually make use of public data to make sense of your own data. And this happens some in proteomics, but we think it could happen more, and we were working on ways to facilitate that. So there have been some efforts to do this kind of thing. So most of the repositories out there provide annotated collections of spectra. So those allow you to search your data against those annotated spectra. And so some examples of those are NIST, Massive, and Pride cluster. In particular, if you look at the Pride repository, this is one of the more widely used repositories. They did a clustering of all Pride spectra in 2015. This is one of the few attempts to actually cluster all of the spectra as opposed to just clustering the spectra for which we know what the generating peptide was. And the challenge here is that the clustering is pretty expensive, and you have to sort of redo it if you want to start over again. So we were trying to figure out a way that we could do something akin to clustering, but that was a little more flexible. And I'm not sure why, but it certainly is the case that this was done in 2015, and there's no evidence it's been done again. Maybe because it's a pretty expensive thing to do. So our concept was maybe we could use the concept of latent embedding to embed all of our spectra into a learned space. If we can use a machine learning approach to embed the spectra into a learned space, then if you stick a new spectrum into that space, you can just see who's close to you. And if they have labels associated with them, then you can adopt their label. So the question is, would this be helpful? Well, ideally, if we do this embedding approach, sure, it might take a while to train it, but once it's trained, it'll hopefully be super fast to just stick new spectra into the embedded space. And so you don't have to relearn the embedding function. All you have to do is put the spectrum in and then look and see who's close to you. So to do this, we needed a way to get spectra into a machine learning system. This turns out to be kind of a pain because of the way spectra, just the way they act. They're not easily summarizable as a simple vector. They're really bags of peaks. There's where each peak is a mass to charge ratio and an intensity, and the mass to charge ratios have to be represented quite precisely. And there's also a precursor mass, which is the mass of the intact peptide divided by the charge. And that's also important, actually very important. So we spent a while, and I'm not going to go through much detail, thinking about different ways to represent spectra or input to a machine learning system. And we came up with this, where you have a couple of experimental parameters that are provided as input. So the mass accuracy and the fragment mass accuracy of the device that generated the spectrum. You have peptide information about the intact peptide, and then you have fragment information about what the locations are of all the peaks in the spectrum. And so these three pieces of information Oh, and I should mention, we also use similarity to a collection of reference spectra, where we basically use the pattern of similarity as another set of features to represent the spectrum. And then those three sets of features are each processed in a deep neural network, where they each get processed separately through some layers and then jointly thereafter. So many of you are probably familiar with these kind of terms from deep neural networks, but just in case you're not, you have some convolutional layers, which are designed to sort of scan across a region of data, or a data object, and find local patterns therein. And then you have fully connected layers that take a bunch of information and consider it jointly. And so this is a picture. We have 500 reference spectrum similarities. We have the bin fragment intensities, and then we have the precursor features. We handle these convolutionally, the first two sets convolutionally, and then fully connected for the precursor features. Partly because there's no real ordering of the precursor features, so a convolution wouldn't really make sense. And then we combine all of these through another, they're concatenated here, and then combined down here. Again, there's hyperparameters involved. So for example, there's a 32 dimensions is the number of dimensions we chose for the final embedding. There's also sizes for these internal layers and so on. And in the paper, we have some description of how we chose all those hyperparameters. Yeah. So you're talking about whether you're pre-training each of these pieces, and then, or are you talking about how many layers to put in? Yeah. So it's always a question. This is a common, yeah. So the question is, there's a lot more parameters than the ones I just alluded to. Some of them are implicit, like how many layers should I put in, and how many nodes in each layer, and then also, should I do the training piecewise? Should I train part of this first, and what hyperparameters should I use for the learning procedure when it's the hyperparameters of the learning itself? I wouldn't say we cover them exhaustively. We try to give a discussion of that, but it doesn't do justice to everything Damon did of trying things out ahead of time. The nice thing is that we did all of this development on an independent set of data from what we'll report here. So at least we don't have to worry about the results that we report being overfit. But this is a common challenge in the literature, is that it's typical for deep learning development to contain many iterations of trying things out. And to my frustration as a reader, often none of those details are given. I think we did a reasonable job, but there may be probably more detail we could have given, because reviewers will only put up with so much. Yeah. So let me just show you the next slide, because I think that'll answer. So this is what the actual network looks like. You take the previous picture and you stick it in here. There's actually two copies of it. So this is called a Siamese configuration. So there's two copies, but they have the same parameters. This network, and this network have tied parameters. All those convolutions and everything I showed you in the previous slide sit here. They output two 32-dimensional vectors, which we then compute the Euclidean distance between, and we do this kind of contrastive loss on those. So mathematically, what you're seeing here is this is the label on a pair of spectra. +1 means they were generated by the same peptide. -1 means they were generated by different peptides. This is the distance in the embedded space that came out of these two networks. And then this is some pre-specified margin. So the concept is that if these two peptides have the same label and they're farther apart than your pre-specified margin, then this will try to push them together. And if they have different labels and they're closer than that margin, it's going to try to push them apart. Okay? And it just iteratively does that over and over again. This is not something that we developed. This is, I should have the citation on here, from a decade ago. It's very relatively commonly used kind of architecture. Okay, so the first thing that comes out is an embedding. It looks like this if you take the 32 dimensions and you project to 2 dimensions with TSNE. So this is 70,000 embedded spectra. We're colored by precursor mass to charge ratio. You can see that there is some structure there. If you instead color by charge state, you can see a slightly different structure. Question is this useful? So one thing that you can do is zoom in a little bit more closely. So what we did was we randomly chose some of these globs. So each of these things is a little glob here. We randomly chose some of those globs and then looked to see what was in there. Or actually, no, sorry, we did this the other way around. We chose all of the spectra in a randomly chosen charge state and about a 1 Dalton precursor mass bin. And then we colored them one color. And then we randomly chose another charge state and 1 Dalton mass bin and did another color. We didn't do too many colors because you can't see that many colors. But we chose for this one 226 spectra in total, and there's about, 6 or 7 different colors. And what you can see is that this seems to be just the main features, the charge state and the mass, seem to relatively place these things on the plot. So now let's zoom in on this particular one, the one that's here. If we zoom in on it, it actually opens up. You can't really see it. I was going to try to get Damon to send me a better. There's some dots here, which are hard to see, and then these ones over here as well. So what's going on here? Well, if we color those dots according to what peptide generated them, you can see that all of these were generated by the same peptide. These are a mixture of different peptides and a mixture, but you can see some of the same colors show up together. But then the question is, what's going on, or what do we do with this? So there's a number of different possibilities. The first thing we considered was just trying to do some of that kind of grouping that I described previously. Like, can we go find collections of nearby spectra that all belong together? We considered a couple of different ways to do this and ended up with a relatively simple greedy algorithm. So what we do is we find the 1,000 nearest neighbors of each spectrum. So it turns out this you can do quite fast. We use a library from Facebook called FAISS, which is spelled F-A-I-S-S, that allows you to rapidly, you can take these 32 element vectors and take tons of them and find your 1,000 nearest neighbors quite quickly. Then you sort all the spectra, and you sort them first by the neighbor count within some pre-specified distance range tau, and then by mean distance to their neighbors. And then for each spectrum, in order, if it's not already a spoke, you call it a spoke, and you connect it to all of the hubs around it. So here, for example, this guy got connected to a bunch of peptide or spectra that were near it. Then you go and grab the next cluster, and then the next cluster, and so on. And you continue doing that, and then at the very end, sometimes you have to do some combining of adjacent hub and spoke communities if the hubs are too close together. And that's only because the first step was an approximation. You got the 1,000 nearest neighbors, and maybe there's more than that within a distance of tau. So that's usually a very small number of things. So then we wanted to ask, how well does this kind of community generation process work? So we went into test data where we hadn't used this during training, and we asked, how well does it do at making peptides that are sort of pure? They have one peptide in them, and not too many communities with multiple peptides in them. And so you'd like to be in the upper left corner of this kind of plot. If we started by just doing k-means clustering, which seems like a reasonable thing to try, right? Lots of people use k-means for lots of things. Turns out this is really slow. There probably are some speed ups that we could make it go faster, but it's not going to go as fast as the simple nearest neighbor thing that we implemented. But more importantly, it makes too many multi-peptide communities. If you think about it, k nearest neighbor, or most clustering algorithms really are designed to try to cluster things at much larger scale than what we're interested in. We want to find very dense, small clusters. And so this basically puts you way over here, depending on what value of k you use. Whereas this hub and spoke method that we developed works much better. These are just for different values of tau. tau equals 0.12, tau equals 0.095 in that space that we defined using the neural network. We chose the tau equals 0.095 because that means that about 1% of the communities that we came up with are multi-peptide communities. So the nice thing is that this is not that expensive. Embedding 5.6 million spectra takes about 20 minutes. And then the 1,000 nearest neighbors search takes about 7.5 hours for all of those 5.6 million spectra. And then the actual hub and spoke method takes just a few minutes after that. So the question is, does this help us? So what we would like to do ideally is say, okay, we've embedded these spectra. Some of them have labels. Can we now propagate additional labels on top of the ones that we had initially? I'm giving away the answer here, but we get, of the 1.6 million in our data set of 5.6 million that were identified, we can get an additional about 8%. And I'll show you how that happens. So the most obvious thing to do is you say, well, I look at my communities, and if this community contains only peptides with a given, only spectra with a given peptide label or unlabeled spectra, then I can just assign the label to the additional peptides in that community. And that immediately gives us about 40,000 additional spectra. The other thing that we can do is try to characterize what is often called the dark matter of proteomics, which is all of the spectra that you can't identify successfully when you do a standard database search to try to identify it. The things that are the problem in general with this dark matter is that a lot of it is non-peptide junk, right? They're noise or other kinds of contaminants. And so the idea here is that if we can embed this learned peptide embedder and a lot of different spectra all show up near each other, and maybe that will help us to focus in on the ones that are likely to be less junky, right? More likely to be real. And so what we were able to do is then focus on the communities that don't have any label and do what's called a tight target decoy search. So because because of the way the spectra work, we can define a much more fine-grained mass to charge ratio, which reduces the number of candidates you have to search when you do the database search. And so this allows you to essentially reduce your multiple hypothesis burden and get better statistical power. And the result is that by using that information, you get about another 40,000 spectra. And then finally, you can do an open modification search, which means you can essentially allow for lots of different kind of post-translational modifications on the peptides, on the remaining unidentified communities, and that gives you an additional 40,000 spectra. So in the end, you get about, as I mentioned, about, and then finally, that wasn't the final, I forgot how many steps there were. There's an additional step of doing a non-redundant database search to find an additional 5,000 spectra at the end. So that allows us to increase the number of identified spectra by about 8%, which is, if you were considering doing this to something like all of the data in the massive repository as of a couple months ago, that would be about 15.3 million additional spectra identified. And the other thing that we show in the paper, I'm not showing you the evidence here, is that the ones that we can't identify, they're enriched for low quality spectra. So that suggests that perhaps a lot of those remaining ones that we can't identify really are problematic in some way. So that's where we are currently. The future directions for this are numerous. So we obviously, one thing is we've only demonstrated this sort of proof of principle on a relatively small database. So a small set of spectra. So we want to scale up to 100 million, 200 million spectra and really see what happens when we do that. And to see what might happen, we did a downsampling experiment by sort of randomly tossing out some of our spectra. This is not actually retraining the model. We're using a fixed embedding, but just asking if you start making smaller and smaller sets of spectra by tossing out experiments from our repository, what percentage of the spectra don't have any labeled neighbor within the distance of that tau distance that I told you about before. And that seems to be, it's not really surprising. It goes up as you get more and more spectra, right? But the point is, it's not like it's really flattened off here. So the hope is that it continues to go up and we get much more identifications as we get orders of magnitude more spectra going into the database. The other thing that we're curious about is to explore this idea from natural language processing that maybe the latent space encodes some interesting semantics. So word to vec is a common or popular way to do embedding of words in natural language. And this is an example from the word to vec paper where they were able to show that if you embed the word king into a latent space, you subtract the vector for man and you add the vector for woman, you end up near the vector for queen. Right? This is a semantic relationship that's captured by word to vec, similar for lots of superlatives and other participles. So we were hoping that we might be able to see similar kinds of things if we look for example, amino acid substitution or post-translational modifications or even mass changes. And then the longer term idea would be that we could hopefully make this work in conjunction with some of these existing repositories to better enable community-wide services for understanding and interpreting mass spec data as it's generated, so that you could quickly and efficiently upload your data into a repository and get back some feedback about what your experiment resembles, what other experiments resemble it, what peptides you were not able to identify that could be identified, and so on. So that's the whole story, and I think I'm just about out of time, but I'm very grateful that you're here to hear my story, and I'm happy to answer questions. Yes. Yeah. Oh, there's a...
You want to start? Okay, well, since I have it, one question is, so you mentioned that the problem with clustering of spectra is that it's quite expensive to cluster them again as the data sets get bigger. And you mentioned that you may have an advantage here that you can embed spectra using the same model that you develop using smaller data sets. So at what point you do have to retrain your underlying models as you continue adding embedding spectrum?
That's a good question. So I think the answer will have to be determined empirically. I mean, what we would do is have some performance measures of, how accurately the model is recovering known peptides. My hope would be that once you reach a certain threshold, let's imagine you've got 1 billion spectra in there. Probably adding another billion is not going to get you that much more. It's, at a certain point, it's not going to become as important.
So I want to thank you for demonstrating a multi-omics approach to the seminar. We are proud that 12 years ago, 13, 14 years ago, we started here with the National Center for NIH on integrative bioinformatics analysis.
Great.
That's what you demonstrated. I want to ask you specifically about the Universal Spectrum Identifier. Eric Deutsch and colleagues, including the HUPO Protein Standards Initiative, have been working on this. I believe it's ready for publication this year. Are you aware of that? Do you think it's something that would be useful in your work?
I'm aware of Eric Deutsch and HUPO, but not...
It's a search tool,
yeah, no, I'm not.
starting with the spectrum instead of the sequence.
Okay, so it's a de novo search tool.
Right.
Yeah, no, I'm not aware of it. There
Yeah,
are
might
some
be.
others that are coming out. I mean, there have been some recent deep learning methods, for example, and a couple other recent advances in that area. So I'll be interested to see this one.
Great.
Hi. So in your Avocado model, what would cause your model to perform better or worse on one cell type versus another? Is it just the availability of information, or is there some other factor?
So it's a good question. I mean, I think it's a lot of it has to do with availability of data, but it also has to do with not just how much data you have in that cell type, but how much data you have in, or what kinds of data that is. Right? Some of these types of data are more informative than others, but often it's also the case that the cell type axis is not really just a linear axis, right? It's more like a dendrogram. So if you have if you're looking at a blood cell type and you've got 14 other blood cell types, then it's going to be much easier for you because you've got lots of neighbors in the cell type space as well, even if you have relatively small amounts of data. So it is challenging to characterize, like a priori, to say, for this prediction, I think we're going to do well, and for that one, we're not, because we don't really understand some of the structure of that space.
I was wondering if it might be possible to formulate the proteomics task as kind of a regression problem instead of an embedding problem, where you have these spectrum peptide pairs and you just try to predict the peptide directly from the spectrum.
Well, so there's two different questions. I thought you were going one way and then you went another way. So you can definitely, you can frame it as a classification task or a regression if you have a peptide, a spectrum pair, and you want to know how good is this pairing. So the most common way to do it is, and this is what Alexei did first, was to sort of say, here's a peptide and a spectrum. This is a true match. And here's a peptide and a spectrum. This is a false match, right? So that's one way to do it. But I think you were going a different direction.
Yeah, so the way you're using the embedding is you're saying, I want to assign a peptide to this spectrum, so I'm going to look at the assignments of my neighbors in the latent space. But it seems like you could also just try to directly predict the peptide from the spectrum, given a bunch of true assignments.
Oh, I see. So yeah, those are the kind of things that were just alluded to. So you can, for instance, the most recent papers train a deep neural network to say, I'm going to take as input a spectrum and output a peptide. Doing that is hard, right? Because you've got this weird you're a sequence over a 20-dimensional discrete space. So there's some bells and whistles to make that work, but it is possible.
Any other questions? If not, let's. Oh, Brian, you want to?
So the question was, where a project like Encode and 40 nucleome going in the next 5 years? And I would really like to know. We don't know for sure. I think so. 40 nucleome is just in its first phase, and it is what's called a common fund initiative, which means it's funded jointly by the NIH, not associated with a particular institute. And those can only go for 2 phases. So obviously, there's no official announcement yet, but we're 3.5 years in, and we're all hoping that there will be an announcement soon of a second phase. If there is, I would guess that, I mean, it's a 40 nucleome, but it's been very 3D so far. There's not that much time happening. It's a little bit, but I think the next phase will have to be much more time-oriented. It's also, 40 nucleome in particular is focused on imaging and sort of sequencing-based technologies, and getting them to work together has been a big focus of the 40 nucleome, but it's not done yet. It's not really started yet. We're working hard to make that happen, but it's going to run into the next phase. Yeah. Encode is an entirely different beast, right? I think Encode is really mature by now. We're in, phase whatever we're in, 4 or something, and I don't, the initial phase was essentially technology development, the pilot phase. The second phase was a scale up to the whole genome. The third phase was, hey, this is working pretty well. Let's just keep going. It was less clear what the mandate was, and I think that's also true of the fourth phase, except that it's clear that the data has value, right? So moving from cell lines to tissues has been a big push. I think moving toward more integrative kinds of analysis is also a big push. And I think one of the directions that NIH seems to be going is pushing toward better tools for visualizing, understanding, and interpreting the data. So, can you make something, the genome browser was a great thing, right, at the beginning of the Human Genome Project. But we may have moved beyond that. And what other kinds of, what's the blast of the 2020s? What's the way that we're going to interact with the genome in the future in a way that takes into account the fact that we now have 10,000 measurements for every single base pair of the genome. That's right. So a lot of the browsers now allow you to see the 3D structure.
All right. If no more question, let's thank Bill again.